to celebrate his contribution to the IMS. And Ori, where are you? There he is. Anybody doesn't know Ori, I doubt it. He was the director of IMS from 2001 to 2011. And prior to that, he was an associate director. He was the associate director for, I believe, about 10 years. So he's currently professor of surgery and surgeon-in-chief at St. Michael's Hospital. By establishing this event, the Rotstein Lecture not only recognizes your leadership, Ori, but it contributes to the ongoing development of academic life at IMS by inviting preeminent internationally renowned scientists to give a lecture to share and exchange scientific ideas with us. So I'd like to introduce Dr. John Matthay, who's professor of anesthesiology and anesthetist in chief at St. Michael's, who will introduce our guest speaker.
Thank you very much. It gives me great pleasure and privilege to welcome Michael Matthay to Toronto. As you can see from this title slide, we're already envious because Michael is based, he's the professor of medicine and anesthesia at the University of California in San Francisco. He's also the senior associate at the Cardiovascular Research Institute and is the associate director of the ICU at UCSF. Dr. Matthay received his AB from Harvard University in 1969. He received his MD from the University of Pennsylvania School of Medicine in 1973, and did his training in internal medicine at the University of Colorado, and then in pulmonary and critical care medicine at UCSF. He has played a key leadership role in the specialty of critical care medicine, and indeed beyond critical care medicine. As specific examples, he's the former chairman of the respiration section of the American Physiology Society, and he served as a member and then subsequent chair of the NIH Non-Biology and Pathology Society subsection. In terms of awards, Michael has been given a huge number of awards, all of which are richly deserved. He's received from the American Thoracic Society an award for scientific achievement in 2002, which I don't know if they give him more than once, but it feels to me like he's the second round of achievement at this stage. He also won the UCSF Award for Outstanding Work in Clinical Research in 2006, and has a Lifetime Achievement in Mentoring Award from UCSF. In terms of Michael's research contributions, no exaggeration to say that he is the leading researcher in the field of critical care medicine, and that is not an accolade I would give lightly. He's made a sustained and outstanding contribution to our knowledge of the biology of acute lung injury. He has more than 550 publications in PubMed, and you have to keep checking because the number keeps going up. He's a founding member of ARTSNet, which is the premier ARDS clinical research network in the world. And they have made a number of key breakthroughs in regard to how we manage patients with acute respiratory distress syndrome, including demonstrating the protective effects of low-time environment ventilation and optimizing fluid management in these patients. He's been principal investigator on a very large number of clinical trials for acute respiratory distress syndrome. He's a physiologist, and his basic research work is focused on the mechanisms of salt, water, and protein transfer across the alveolar epithelium, both in normal lungs and in health, and studying how fluid is reabsorbed from the alveolus following injury. His recent research has focused on mesenchymal stromal cells and their therapeutic potential for acute lung injury and ARDS. His research group at UCSF has spearheaded the preclinical evaluation of therapeutic potential of these cells, and they have demonstrated efficacy in multiple models of disease, including pneumonia, endotoxin injury, and really exciting today the explanted human lung, which I'm sure we'll hear about today. He is the principal investigator in the first clinical study of mesenchymal stromal cells for patients with ARDS, which is a really exciting program. So the one thing he told me that I had to tell you was he's a lifelong Red Sox fan. That was actually the most important thing I needed to say. But I think nobody is better positioned to tell us about mesenchymal stromal cells and their potential for ARDS. So I am really delighted to invite Michael Matthay to give the 2013 or a Rotstein lecture on the subject of mesenchymal stem stromal cells, biology and therapeutic value for ARDS. Thank you, Michael.
John, that was overly generous. Thank you very much. Alan and Maury, it's a delight to be here. Ring me out. Thank you very much for inviting me. And it is an honor to be here. I consider the University of Toronto and your many locations here really a mecca, and very pleased to be here. Now, I'll feel particularly at home if you interrupt me along the way here, because we all know at home base our colleagues and friends and fellows interrupt us. So even though there's a certain formality to this lecture format, please interrupt me during the course for any issues that aren't clear. I think there are several fellows here, so please just stop me along the way. You just see here, and then to advance, I just do this. Okay, so first and foremost, in terms of the title, I think this is an area that Armand, where's Armand? Right there. So we probably discussed this issue. We try to escape from it by, from here on out, the rest of the talk saying MSCs, but in fact, as I think many of you know, stromal is in many ways a more correct term. The stem properties of the MSCs is more limited. So that the stromal here, I think, is important. And so I'll just, let's see, I can advance that. We're advancing.
Help is on the way.
Show me which one I should use.
Canadian version of the keyboard.
Oh, perfect. All right, just this. Oh, that's great. Thank you very much. So just a disclosure slide, no conflicts. The work I will show you is all supported by the NIH through some basic grants and then a translational grant that I'll tell you more about for our phase 1-2 trials that are just beginning with, and we're including Stanford, MGH, Pittsburgh, and Vermont in these studies. And as I'll also tell you about, we depended very much on a NIH repository at the University of Minnesota. Like most research, there's always different stories that are relevant for how the work was able to go forward, and this is an interesting story I'll tell you a little bit more about. So my plan for today is to give a very brief historical perspective on mesenchymal stem cells, or mesenchymal stromal cells, MSCs, say a word about ARDS and pulmonary edema, an area of expertise here for you in this area, so I don't need to elaborate on it. And then I'll go through the preclinical studies we did first in mice, and then the ones we have done and are still doing in ex vivo perfused human lung, as well as some sheep experiments that form the basis for our efforts to translate and test MSCs in the clinical setting, and tell you a little bit about that trial, and then some of the many unanswered questions in the field. So I think you probably know this, but for those of you who do not, Dr. Friedenstein, a Russian scientist, is credited with having discovered or first identified bone marrow-derived mesenchymal stromal cells. Here is an image of MSCs. When you look at it, they really look like fibroblasts. There's nothing about it that would make you think they're anything other than fibroblasts. And the story is that the Russian government required that Dr. Friedenstein isolate bone marrow from donors every Thursday or Friday and save this bone marrow, freeze it in the case of a nuclear war with the U.S. or a radiation disaster like Chernobyl. And so he did what they told him to do. He had to. And in the process, he noticed initially that his isolates, after he had removed the supernatant, where the bone marrow hematopoietic elements were, that the plate was covered with cells that looked like this. So he thought he had fibroblast contamination. But after a while, he realized, no, these are probably stromal cells from the bone marrow. And like with many discoveries, there was a quiet period for many years before scientists began to investigate them. And then starting in the late 80s and in the 90s, several immunologists and hematologists began to study some of their immunomodulatory capacity. Now, in the field of pulmonary disease, Diane Krause at Yale in 2001 published a paper in Cell in which she thought there was evidence that these bone marrow-derived MSCs could graft into the mouse lung and actually form alveolar epithelial cells. This was actually a potentially major finding. However, she, as well as other investigators in Boston, fairly shortly thereafter, figured out that in fact that was not the case, and they've been misled a little bit by some of the staining and some artifacts. And so there really wasn't evidence that these cells significantly engraft in the lung. However, like so many articles that might be a little misleading, and still it stimulated investigators to study MSCs, and many investigators began to focus much more on their paracrine properties and how they could modulate cell and organ function. Now, this slide is intended to just remind you that these cells isolated from the bone marrow have some properties that can be reproduced in vitro to identify that, in fact, they are mesenchymal stromal cells. They can be induced to form osteoblasts or adipocytes or myofibroblasts under specific in vitro conditions. In terms of their surface markers, they're negative for CD11 and CD45, and CD34 and CD31, and positive for CD73, 90, and 105. The field is somewhat plagued by the fact there isn't a single marker that absolutely defines these cells, but that's about where we are now. And there's a lot more details in that regard as well. And these cells have been isolated, as most of you know, from bone marrow, adipose tissue, umbilical cord, and many other organs. Now, this recent review in Nature Medicine summarizes very nicely a lot of the work that's been done in evaluating and studying the interactions of MSCs with several different cells. Almost all you can imagine here: B cells, T cells, monocytes, macrophages, neutrophils, as well as endothelial and epithelial cells. It's quite a story. It's very interesting, and there's a growing literature on multiple levels regarding these cells. We are also studying placental-derived cells, and I think there's just an enormous amount to be learned about the basic biology, apart from any clinical applications. Now, our interest from a clinical scientific perspective, in part, has been focused on non-cardiogenic pulmonary edema, as shown in this slide, or in this X-ray, in this histology here, or what we call acute respiratory distress syndrome, ARDS. As you know, ARDS is a frequent problem. In the U.S., it appears there are about 200,000 ventilated patients annually with mortality of about 20 to 35%. And the primary causes are bacterial and viral pneumonia, sepsis, shock, particularly after major trauma, and multiple transfusions, and aspiration. Now, supportive treatment with lung-protective ventilation has had a remarkable effect on reducing mortality in this syndrome. It's an amazing story, as you know. Probably dropped mortality close to 50% of what it used to be. And a fluid conservative strategy has benefits in terms of patients getting off the ventilator sooner than they used to. But we still lack a more specific therapy that would, one, reduce the severity of lung injury initially, and two, perhaps hasten recovery. And that forms the basis for our interest in MSCs. So let me take you through just a few slides showing the early work we did with mesenchymal stem cells and mouse models of lung injury, and then I'll go forward from there. So this slide shows on the y-axis 48-hour survival, and this shows the time frame in mice that were given very high dose of endotoxin, 5 milligrams intratracheal. And then 4 hours later, the mice were treated either with bone marrow-derived MSCs, which we had derived ourselves, or also we obtained some from Darwin-Procope's repository. And controls with PBS. Now, there are additional controls we did, including 323 fibroblasts and apoptotic MSCs. And the major finding you can see here is that the MSC-treated mice had a much better survival. They look completely different. And in this case, we gave the MSCs intratracheal 4 hours after the injury. We found at that time, I thought it was important because we're using endotoxin. In fact, MSCs express TLR4 at the gene and protein level, but the studies that showed that the MSCs were not working simply by scavenging the LPS, which was a possibility early on. These experiments showed that the treated mice had a significantly lower extravascular lung water. This is a pretty big effect because normally there's a lung water of about 4 grams per gram of dry weight. So this is a more than 50% reduction, showing the same data graphed as excess lung water. And if we lavage the mice at the end of the experiment, the quantity of protein in the airspace is reduced, indicating there's less exudate. Histologically, the lungs were much different in the treated mice. As you can see, here's the PBS controls with consolidation, edema, neutrophils, and this was supported by semi-quantitative scoring. And then we also, in that first experiment, as expected, found that the levels of pro-inflammatory factors were reduced in the air spaces and in the plasma. In this case, MIB2 in the plasma and the BAL and TNF-alpha, which didn't quite reach significance. There was also an increase in anti-inflammatory cytokines, including IL-13 and IL-1RA. Now, the big question then was, okay, this works with endotoxin, but how would these cells work in the presence of live bacteria? In fact, my fellow who was working on this project at the time, Dr. Gupta, he didn't want to do the experiments. He said, well, Michael, they're not going to work. These are immunomodulating. It's going to make it worse. I said, well, it may be, but we have to do the experiments. You know, we'll have to see. And so he did the experiments, and he was quite surprised to find there was a very similar effect as with the endotoxin. We used our live bacterial model with E. coli, same design where we gave high doses of E. coli into the trachea, and then followed 4 hours later with intratracheal mesenchymal stem cells. And you can see here that the survival in the dotted line for the MSCs was much better than the fibroblast controls of the MSCs. He didn't really believe it. He says, I can't believe this. And so actually we said, okay, we'll blind you. Let's do another set. So we did it again. It kept coming out this way. And in experiments that we have done subsequently that in the interest of time I didn't show you, we've also done this not just with mouse bone marrow-derived MSCs, but with human MSCs. They do not express several antigens that would make them rejected, and so you can actually give human MSCs in short-term experiments in mice, let alone in people, something we'll come to. And then, but the major unexpected finding here, other than the efficacy, was that the number of bacteria that grew out of the lungs was markedly reduced in the MSC-treated mice. So this was quite unexpected. We didn't expect an antimicrobial effect. As I mentioned, we actually anticipated the opposite. This is data showing at 8 hours the number of E. coli in the bronchoalveolar lavage of the MSCs versus 3T3 and PBS controls, and this shows in the number in the lung homogeny. So this began a search for trying to figure out how this was occurring. And it was particularly unexpected. And I'm going to try and explain this briefly, but I'll come back to it also when we discuss the human lungs. But what we found after searching for several factors, and because we were also advantaged by a Russian scientist, Dr. Krasnodemskaya, who came from St. Petersburg to work with us, a woman who had cloned some antimicrobial factors in worms as part of her PhD thesis. So she understood antimicrobial factors certainly better than I did at the time. And it turns out that MSCs release themselves and induce from other cells the antimicrobial factor LL37, beta-cathelicidin. And in these experiments here, you can see that when we gave a neutralizing antibody to LL37, in this case, these were human MSCs, it blocked the growth of the bacteria. This is in the lung homogenate, this is in the BAL, and this is an in vitro setting. And we also found with mouse MSCs that they release lipocalin-2, which is an antimicrobial factor. So it became apparent that these cells had antimicrobial defenses that was part of their properties. We did one other set of experiments with infection other than just pneumonia. These were studies done with bacterial peritonitis in which we gave high-dose Pseudomonas into the peritoneum with the idea of trying to test how MSCs would or would not affect host defense in the face of a very acute lethal bacterial load. And you can see here that in these mice, they were given a high-dose Pseudomonas into the peritoneum, and then 1 hour later were given human MSCs by a tail vein, that in fact the mortality was better in green with the MSCs compared to 323 fibroblasts or PBS. And again, in terms of the bacteriology, the number of bacteria that could be cultured was significantly less than the MSC when we measured it from the blood. There's only one that we had measurable bacteria from compared to the other controls, and the concentration, the number of bacteria in the spleen from these animals was different at a p equal to 0.05 level. So we, in a very acute experiment, found that again there was the antimicrobial effect. And in this course of doing these experiments, we learned not only were there antimicrobial factors that are elaborated by the MSCs or induced, but that in addition, it appeared that the MSCs enhanced monocyte phagocytosis in bacteria. We tested neutrophils, and there was no change in neutrophil phagocytosis. But in all the experiments here in the mice, we found that phagocytosis was markedly increased. Now, this observation has been confirmed also by Duncan Stewart's group with some work that Dr. May did. And so we now had two pathways, and I'll come back to this when I tell you about the studies in the human lung. And then the one other observation to mention from the mouse studies is that it appeared that the MSCs upregulated the expression of two markers of alternatively activated macrophages in the spleen. You can see the CD163 and CD206 in the MSC-treated mice in the spleen. And this was consistent with some other work that suggested that the MSCs seem to stimulate the M1 monocytes, the pro-inflammatory ones, to a more anti-inflammatory phenotype. And it looks like C5a was higher in those mice who had this property, but we and others are still working on the mechanism by which this occurs. But obviously this could be important in terms of understanding how the MSCs might enhance resolution. So just to summarize the results of the studies in the mice, intratracheal or intravenous mouse MSCs or human improved survival and reduced lung injury in both endotoxin and live bacterial models of lung injury, both associated with a reduction in pro-inflammatory responses, an increase in anti-inflammatory cytokines, and apparent reprogramming of M1 to M2 monocytes. And the antibacterial effect of MSCs in mice was mediated by antimicrobial peptides and enhanced monocyte phagocytosis. Now, of course, we are certainly not the only investigators working in this field, and this is a incomplete list of work that's been done also recently that I think is important. Luis Ortiz, in a PNAS paper in 2007, reported that MSCs reduced bleomycin-induced lung injury, largely by IL-1RA dependent pathways. Nemeth had a very nice paper in Nature Medicine in 2009 showing that MSCs reduced cecal ligation sepsis mortality, apparently in part by upregulating macrophage anti-inflammatory properties. They didn't study the antimicrobial part of the issue as much as we and Dr. May's group have. And then Gerard Curley, here with John Laffey, published a very important paper from work they did in which they showed in rats that MSCs reduced ventilator-induced lung injury in rats, and importantly, they did this with 2 doses in over 48 hours. In other words, it wasn't just a acute intervention model. And then the nice work from Dr. Duncan Stewart's group showing that MSCs reduced cecal ligation-induced sepsis mortality in mice and they had some very good data on how monocyte phagocytosis appeared to be upregulated in the spleen. So let me move on to the human lung studies and tell you a little bit about these. So for over a decade, our research group has been fortunate to have a very close relationship to the Northern California Transplant Donor Network. Now this is the organ procurement group for Northern California. It serves about almost 10 million people. And this is the group that is called in when a patient is brain dead and then takes over the management of the brain dead subject and then evaluates the subject for the possibility that their kidneys, liver, heart, lungs, and so on can be transplanted. So for more than 10 years, we have had a very collegiate relationship with them. So they send us their lungs that they are not being used for transplant. So this has been a great help to our program. We've studied mechanisms, as John had mentioned, of alveolar fluid clearance, how it is that the epithelium can remove edema fluid from the lungs. So it's been a big help to have these lungs. We also isolate alveolar epithelial type 2 cells from the lungs, and we've shared these lungs with, of course, other investigators at UCSF and other groups. Now, for these particular studies, what we did, starting back in about 2005, is we took the lung, and it comes to us at 4 degrees, and we cannulate the trachea and the pulmonary artery, and then we rewarm it slowly with a physiologic solution up to 37 degrees by perfusing it. Left atrium is open. It's in a perfusive reservoir. It's a recycle here, just a simple Harvard pump. Then, in all our experiments to date, with a couple exceptions, we've kept the lung inflated with CPAP, 10 centimeters of water, 95% oxygen, 5% CO2. And we run the flow so that the PA pressure does not exceed 10 to 12 millimeters of mercury. And that way, there is not, the lungs are quite stable and don't accumulate edema significantly. So the original study on this was made it possible for us to measure alveolar fluid clearance in the human lung, which simply means how well is alveolar edema removed from the airspace of the lung. And those lungs that are reasonably normal, we found that it's removed at about 20% per hour. Now, how does alveolar edema fluid clearance occur? What drives it? Well, it's driven by sodium chloride transport, active ion transport from the type 1 and type 2 epithelium. The ions are transported to the interstitium and draw water across. So that's how edema fluid clearance occurs. And in a normal lung, it can be upregulated with beta agonist, so the clearance actually doubles to 40% per hour. So we adapted this preparation in order to try and test human mesenchymal stem cells. Now, for these experiments, we tried to take lungs that looked and appeared to be normal, not ones that were injured by trauma or had consolidation or widespread atelectasis. And I'll come back to this more. And so again, we initially set up the lungs, perfused them, and then we did one other important procedure here. We took the right upper lobe, we measured one lung. I should have made it clear, we used one lung for these studies, not both lungs. And we measured alveolar fluid clearance in the right upper lobe. How do we do this? Well, we take a solution of 5% albumin, distill it into the right upper lobe, and if fluid clearance is intact, the protein poor portion of the insulin will be reabsorbed. So if the albumin concentration is 5 grams per 100 ml initially, it'll go up to 7, 6.5 grams over 1 hour. And so we use that as a test to convince ourselves that these lungs are normal, not just the way they look, but in fact by this test, because we want normal lungs to start with because we're going to injure them, at least for these experiments. Then, through a process of trial and error, we decided to add to the perfusate fresh whole blood. So one of us gives about 150 mLs of blood. So we have red cells, neutrophils, monocytes, and platelets in the circuit. It gives hematocrit about 3 to 5%. And then for these experiments, initial ones I'll show you, we instilled 6 milligrams of endotoxin, very high dose, into the right middle lobe. And then treated the lungs 1 hour later with either intratracheal or, as I will show you, intravenous in the perfusate MSCs or appropriate controls, namely fibroblasts. And then the initial experiments were 4 hours. During this time period, as I will show you, we measured vascular permeability, lung weight gain, and at the end, alveolar fluid clearance. So this slide shows the results of our initial experiments in which Dr. Lee measured endothelial permeability. We did this by putting a radio-labeled tracer, I-131 albumin, into the perfusate and then measuring its accumulation outside the vascular compartment. And you can see with endotoxin that vascular permeability went up 3-fold. However, remarkably, giving the MSCs or conditioned media of the cultured MSCs, vascular permeability returned to normal. When we did the wet to dry ratio as an index of pulmonary edema, at the end of the experiment, controls were here at about 5. LPS was up here at year 7. And you can see with MSCs it returned it to normal, and with conditioned media even slightly less than normal. And then what about alveolar fluid clearance? Well, as I mentioned, in normal lungs, alveolar fluid clearance is about 20% per hour. Here's the endotoxin, completely obliterates alveolar fluid clearance. And what's very interesting about this is that I'll show you in a few slides some work from Dr. Bhattacharya in New York. Johar showed that when he put the same dose of endotoxin he used in the mice into their lungs, surfactant secretion stopped, just immediately stopped, almost exactly like this. No alveolar fluid clearance, no surfactant secretion. Treatment with normal lung fibroblasts did not change alveolar fluid clearance, but treatment, as you can see with MSCs or the conditioned media, 1 hour after the endotoxin did restore transport to a normal level. So naturally, we tried to search and understand what were the mechanisms that mediated this. And in the interest of time, I won't give you the whole story, but we did an siRNA knockdown for KGF, an important fibroblast growth factor that we and others have reported has beneficial effects on the injured lung. And when we measured alveolar fluid clearance again, and then we gave conditioned media in which the KGF had been depleted with an siRNA strategy, you can see that about 70% of the beneficial effect was lost. And then when we gave recombinant KGF, shown in yellow here, alveolar fluid clearance returned to normal. So part of the beneficial effect in these experiments was from KGF. We have other studies with, for example, isolated type 2 cells in monolayers in which we increased permeability with cytomix. In those experiments, angiopoietin-1, elaborated by the MSC, is the important factor in restoring permeability. Okay, so at that point in time, here's where we were. We began to think about the possibility of trying to translate this work to patients, but there were several barriers to translating human mesenchymal stem cell therapy to patients. Number one, we didn't have a reliable source of high-quality clinical-grade human MSCs. We had been obtaining these MSCs from the NIH repository of Dr. Prokop, but they weren't clinical grade. Secondly, we didn't have funding. And 3, although we have been a major center for NHLBI-sponsored phase 2 and 3 clinical trials since 1995, we did not have the expertise at UCSF and in our group for how to know how we would proceed with an IND, approval to the American FDA, and the steps to testing the mesenchymal stem cells in the clinical setting. And I was searching around for companies or other groups that might provide the cells, and for a variety of reasons, none of that was very promising. And then I was very fortunate because the investigator at UCSF said, "Oh, Michael, don't you know about the PACT program?" I said, "What's the PACT program?" And they said, "Well, this is a program funded by the NIH, and the lung division of the NIH is in National Heart, Lung, and Blood. So blood is part of this division. But like many places that get too big, they don't always talk to each other. And although the lung division people knew we were looking for a clinical source, they never told me. But somebody at my place said, no, look, at the University of Minnesota, they're one of the PAC groups. Their job, their main purpose is to produce cells that have clinical grade qualities and can be used for a variety of trials. They're involved in hematology and other trials as well. So I was able to connect with Dr. Dave McKenna, the head of this program, and he had basically had isolated and expanded bone marrow-derived human MSCs, and were wishing they had a contact with someone who could help translate this. So from that day forward, all of the subsequent experiments virtually we've done, almost all of them, have been done with clinical grade human mesenchymal stem cells, the same ones that we would give to patients, with the idea that we really test them in the human lung and other settings I'll show you. So, then, having done the endotoxin studies in the human lung, and now having clinical grade human bone marrow-derived MSCs, we then challenged the human lung with live bacteria. And we also wanted to see if the MSCs would work by the perfusate, not just by giving them in the trachea. So here on this slide, I'm showing you alveolar fluid clearance, and I'm showing you control, the E. coli, live E. coli, large, very large inoculum, and then here intrabronchial or intravenous MSCs. And so this was an attempt to now test whether the lungs would respond to the MSCs as we'd seen in the endotoxin or in the mouse studies. You can see alveolar fluid clearance returned about 2/3 of normal. And interestingly, to my surprise, look at the absolute neutrophil counts in these experiments on the y-axis compared to the x-axis here. Here's control in the alveolar compartment after 4 hours. The number of neutrophils goes way up, of course, with the bacteria. But with intrabronchial or intravenous MSCs, again given 1 hour afterwards, the number of neutrophils is way down. In this slide, I'm showing you bacterial counts on the y-axis to see if we found the same antibacterial effect we had in the mice. And you can see that the E. coli alone here, and intrabronchial reduced them dramatically, and intravenous reduced them too, and when we doubled the dose of introduced MSCs, you can see it reduced the number of bacteria further. And this was some in vitro experiments we did also that showed the same pattern. So we now found a antimicrobial effect like we found in mice. Because of this prior finding that there was enhanced monocyte phagocytosis in the mice, we also studied this pathway in the human lung, and indeed found that with giving the MSCs, there was a marked increase in monocyte phagocytosis in these experiments, no change in neutrophil phagocytosis. So this finding appeared to be relevant to the human lung, not just the mouse lung. And now when we submitted the manuscript for review, the reviewers came back and said, well, it's interesting, but can you do some more? Why don't you give antibiotics? We want to see what antibiotics do in this setting, and we'd like you to make the experiment longer. Well, it's hard to keep the human lung without a bioreactor in good shape for a long time. We actually increased the experiments to 10 hours, and we also gave the MSCs 2 hours after the E. coli instead of just 1 hour, and we increased the number of bacteria by a whole log fold, 10 to the 10th. It almost killed Dr. Lee, but he has survived. And here's the data. I want to show you on this in the next slide. It was, I think, quite interesting. So here's the basic experiment. Here's the control, alveolar fluid clearance. Here's the E. coli where the clearance goes way down. Here's intrabronchial MSCs, which brings it back up toward normal. And here's IV ampicillin. Ampicillin killed E. coli very effectively. It was an ampicillin-sensitive strain, but did nothing to alveolar fluid clearance. And here's the combination where you needed, apparently, the intrabronchial MSCs to enhance fluid clearance as well as the antibiotic. And then the next slide I thought was quite interesting because we also cultured the perfusate as well as the alveolar compartment and homogenate. And what we found is that when we measured the E. coli in the perfusate, so an indicator that the bacteria translocated from the airspace into the perfusate, this shows you the data for the control for the E. coli without treatment. Here's the intrabronchial MSCs. Notice with the MSCs alone, there's no bacterial translocation to the vascular compartment. No antibiotics, just the MSCs, suggesting that either the antimicrobial effect or perhaps a tightening of the epithelial barrier, both is important And of course, if we gave IV ampicillin, we eliminated translocation also. But the MSCs alone were able to decrease bacterial translocation in this model. So this slide is intended just to summarize these human lung experiments that which we use the human MSCs. It just shows spot histology, which is open to bias because these were done with blinding. But here's the control of lung. Here's the lung with E. coli pneumonia with lots of consolidation injury. Here's the normal human lung fibroblasts, no difference. And here's IT or IV MSCs or IV MSCs double dose. And you can see it's quite different. So one, the human MSCs reduce lung endothelial and epithelial injury. They increase the resolution of alveolar edema, enhance bacterial killing, and reduce bacterial translocation. And in data I haven't shown you enough of, KGF has mediated some of these beneficial effects. In the interest of time, I couldn't show you, but in these human lung studies, we actually found that monocytes express the KGF receptor, a major surprise. And the KGF stimulates the monocyte uptake of bacteria through this receptor. There's another group that studied KGF in a different experimental setting, also found antibacterial effects. So KGF started out as an epithelial-specific receptor, but it's not. It's on the lung endothelium in a major JCI paper about 3 years ago, and I think this data shows in our paper that it's on monocytes. And then I put in one slide. I wanted to show this particularly for Shaf. Shaf, where are you? Clear. Good. So maybe we could go into this in more detail. Gerard Curley did these experiments with Danny McCauley and Jay Lee at our place, and these are experiments where we took, instead of taking lungs that were normal, we now took lungs that were injured. So these were lungs that definitely would not be transplanted. And when we measured alveolar fluid clearance in that initial phase, like I mentioned, after we warmed them up, there was no alveolar fluid clearance. So they wouldn't have made it in these experiments that we did where we actually injured the lung. But I wanted to show you that when we took these lungs, these are experiments not yet published, but baseline clearance was very low. So there's something wrong with the alveolar epithelium. And we then gave them intravenous mesenchymal stem cells. And you can see that it increased alveolar fluid clearance to almost a normal level over 4 hours. Perfusion alone had no effect. And not shown here, but if we have time we can go into later, we give a neutralizing antibody to KGF. It takes away some of this effect. So this speaks to the issue that possibly some of these injured lungs can be rescued. Of course, the sentinel work on this is Schaaf's New England Journal of Medicine paper with the in vitro life studies. But I think this shows that there's a potential application here. And so I just put it in because I thought it was relevant.
Right.
Yes.
Why do you think that we see some of the perfusion stimulation effect of fluid clearance
I think because these were pretty injured lungs. I mean, we didn't take ones that were, severely consolidated. I mean, really major consolidation or traumatic injury. But through the process of whatever happened in the brain dead organ or during the harvesting, they did not have fluid clearance. So perfusion alone wouldn't bring them back to normal.
I think in our clinical experience now, we're beginning to see that, and we're seeing improvement beyond what you'd expect.
Just with the perfusion.
Just with perfusion. So.
Well, the thing is that we didn't do it in as comprehensive a way as you've done. We didn't ventilate the lungs like in the vitro life. We were just perfusing them and putting them on CPAP. So we really didn't maximize. We didn't have a steam solution. We just used the 5% albumin. In these perfusion-only experiments, we did not use blood. So we probably did not maximize the chance that just better perfusion and ventilation would improve their function. I think that's absolutely right. We only went for 4 hours. So this slide is intended just to summarize briefly that there are several paracrine factors that our group and other investigators identified that seem to be important in explaining some of the beneficial effects of MSCs in the lung that's acutely injured, whether they're given by the air spaces or intravenously. KGF certainly seems to be important in our experiments. Ang1 and data I didn't show you is important. And the interesting thing about Ang1 is prior work is primarily focused on its effects on the endothelium. That Ang1 is the counterbalancing for the Ang2 induced permeability effects. But we found it works very well on the epithelium. In addition, there's the IL-1RA data I mentioned from Dr. Ortiz, the 2 papers. And of course, in the Nemeth paper, they thought PGE2 was important as a mediator of changing the macrophage to an anti-inflammatory phenotype. And there are pathways that are known and not known that are important, that are affecting monocytes, like I described for increased phagocytosis, and a lot of different interactions. But clearly, there are paracrine molecules that are working. However, I think the story is not just about paracrine effects. Excuse me. This isn't coming out as I would like, but that's okay. I can tell you the story. So this is this slide and the next one from Dr. Islam and Bhattacharya's paper in Nature Medicine last spring, or in 2012, in which Jahar and he had used the same model of endotoxin injury that we had in the mice. And as I mentioned, he found that surfactant secretion was stopped. And then when he gave MSCs, surfactant secretion was completely recovered within a short time period. Well, the major point of this paper was that it appeared that the MSCs transferred their mitochondria to the injured alveolar epithelium. And I can't show you too well because the slide didn't come out right, but this is a terrific paper. He showed with human MSCs, for sure, using human mitochondrial DNA, that in fact it was the MSCs that had transferred the mitochondria to the injured alveolar epithelium. And then through some very clever studies, he showed this depended on connexin 43. The pathway for the transfer of the mitochondria was not clear. It's not through the connexin 43 bridges, or probably through some process of endocytosis. And the work suggested, let's see what happens here. The work suggested, and their hypothesis, well, it won't come through completely, but that the benefit was explained in part by restoration of ATP levels, bioenergetics, if you will, to a more normal level in the injured alveolar epithelial cell. So this model would say that it depends on cell contact, and that the cells themselves are important for transporting perhaps even subcellular organelles, not just the paracrine factors. And this slide is trying to summarize those 2 ideas. On the left here is the MSC, in this case diagrammatically releasing Ang1 or KGF to the epithelial type 1 cell or type 2 cell. And on this side, based on Jahar's work, is the MSC with connexin 43 bridges releasing mitochondria into the epithelial cells. This may sound like voodoo science, but it was actually Darwin Crockett in the PNAS paper who published before that evidence that the MSCs do give up their mitochondria in culture to epithelial cells. So how this occurs, how important this pathway is, there's a great deal to be learned, but at least creates some equipoise in the field about the mechanisms. Okay, so finally, let me just show you some experiments we did at sheep. When we worked with the FDA, they were positive about the plans to translate this. They were happy with the source of the human MSCs from the NIH repository, and they were reasonably pleased with the preclinical mouse and some other rat studies we did in the human lung. But they said, you know, it would be much better, don't you think, Dr. Matthay, if you had a large animal model? And I said, oh yes, you're probably right. And I actually, the truth is that I tried to talk them into rats only. I said, well, we do rat studies very well. We put the arterial catheter in the carotid. We can measure airway pressures. We pointed to, Gerard's and Jones' paper. And they said, well, if you think that's adequate, but, the better side of me emerged there a little bit. And I said, well, maybe we could do some sheep studies. I had originally began my research career working with sheep and alveolar fluid clearance. But we don't have facilities for sheep work at UCSF. So luckily, I was able to collaborate with Dr. Dan Traber and his wife, who's an ICU nurse, and their whole crew at the University of Texas in Galveston, at a terrific sheep facility for a variety of sepsis study and acute lung injury stories. It's very sad. Both Dr. Traber and his wife passed away, and so we're left with Dr. Inkabakhtar. Somehow we got all these experiments done. Dr. Dan was just like a hero. He, even as sick as he was with the metastatic cancer, said, "Don't worry, we're going to get these done, Michael. They're too important." So it's because of them we got these studies done. Now here's the story. Severe acute lung injury in an anesthetized sheep injured by inhalation of cottonwood smoke, hot smoke, plus installation of live Pseudomonas aeruginosa into 3 lobes, followed by mechanical ventilation, done in an anesthetized sheep, as in patients with ARDS. The sheep get very severe lung injury. The week before, the sheep were prepared to measure pulmonary and systemic hemodynamics, respiratory function, urine output, and we, in all these experiments, collected a large number of biochemical indices throughout the experiment. The design was 24-hour experiments with a primary focus on safety, administering the MSCs 1 hour after the injury. The plan was to use 5 or 10 times 10 to the 6 cells per kilogram, plus our efficacy endpoints. In terms of the issue of safety, you can imagine the risk here is that you give these cells, even though it's not a huge number and it's 1 order lower than you're given a bone marrow transplant, that we run the risk of obstructing the injured pulmonary microcirculation and create transiently right heart failure. So here I'll just show you 3 or 4 slides from the experiments. The control for plasma-like alone, that was the carrier, and then the lower dose MSCs 5 times 10 to the 6, and the higher dose 10 times 10 to the 6 per kilogram. And in this slide I'm showing you systemic arterial pressure. And in fact, there is no deleterious effect. Here's the injury being given, the smoke and the Pseudomonas. You see the blood pressure goes up, and we have cardiac output and systemic vascular resistance. It all went up, and the SVR down. But you can see that, in fact, at the end of the 24 hours, statistically, the blood pressure is actually higher in the MSC-treated sheep. Now, that wasn't true for cardiac output or systemic vascular resistance, but there was no apparent deleterious effect, which was reassuring. These are very sick sheep. 3 people with a sheep, 24 hours, collecting lactate every 3 hours, all the chemistries, hematocrit, and so on. And here's the pulmonary arterial pressure. And again, blue for control, green the lower dose, and red the higher dose. And so what we were concerned about would be the increase in pulmonary arterial pressure that was greater in the treated mice. The treatment was given 1 hour after the injury, over a 1 hour period. And again, you can see that actually it appears that the sheep that were given the higher dose has statistically lower PA pressure, probably not clinically significant, but the main point is it didn't go the wrong direction. And that was, and the rest of the safety parameters were reasonable. Now, what about efficacy? Well, we measured the oxygenation. Shown here is the PaO2 FiO2 ratio. All these sheep are being ventilated. The fluid protocol is according to a protocol in which the hematocrit is to be maintained within 3% of its baseline. And what you see here is that the plasma-like treated mice, notice that their oxygenation plummets. And so within 2 to 3 hours, they're down to PF ratio about 200. Remember, for clinical purposes, a PF less than 300 is ARDS. These are very sick, and watch here, you can see they go out to 24 hours, and they have a PF ratio less than 100. The MSC-treated sheep, if you take, we did statistics on this as an area under the curve approach, and it is, there is statistically better oxygenation in the higher treated MSC group, the ones in red, and both of them at 24 hours. Consolidated. The lower dose did not reduce lung water, but the higher dose did. And so this is the dose for our clinical trial that we're hoping to get to based on this data. So, so here is a brief description of the clinical trial. The goal is to test allogeneic bone marrow-derived human MSCs from the University of Minnesota repository in a clinical phase 1 and 2 trial of severe ARDS, so a PF ratio less than 200, with at least 8 centimeters of water peak. So we don't want to take patients with just mild ARDS. That would seem inappropriate. We are focusing the trial on safety and respiratory efficacy endpoints such as oxygenation and pulmonary dead space fraction. And the design is to test potential biologic pathways of benefit by sampling the plasma and the airspace of the lung. And as I mentioned, this is funded by the NHLBI. We have begun the phase 1, and we've enrolled 3 patients at the lower dose, which was 1 million cells per kilogram, and now we're moving on to test 5 million cells per kilogram for 3 more patients. If there's no safety issues there, we'll go on to 3 more patients at the target dose of 10 million cells, and then to get a phase 2 design of 60 patients with 40 treated and 20 controls. It's randomized and double-blind. So how might, in summary, mesenchymal stem cells work in the presence of acute lung injury and ARDS? Well, we know that even if the injury is initially from the airspace compartment, as in aspiration pneumonia, that the increased vascular permeability is the primary lesion in this syndrome, so that the capillaries begin to leak protein-rich fluid mediated by neutrophils and platelets and other factors. Interstitial edema forms. The fluid translocates to the alveolus to make protein-rich edema in the air spaces, which is what impairs oxygenation. The normal epithelial cells shown on this side, the type 1 cell, the type 2 cell that makes surfactant, is often very injured. In fact, it's often not present in the more severely injured alveoli. You just see a hyaline membrane. In other areas, they're there, but partially injured. And so we have a combination of endothelial injury, epithelial injury, procoagulant properties, and an intense pro-inflammatory environment. So with the MSC hypothesis, our hope is the anti-inflammatory properties will be beneficial. It appears that they have the capacity to restore endothelial and epithelial barrier integrity. It appears they can enhance alveolar and lung edema fluid clearance. They do have antimicrobial properties. There is evidence I haven't shown you from our group and others that they have anti-apoptotic effects. One of the interesting discoveries in the field is some very fine Italian investigators reported that in ischemic renal failure, MSCs or even the microvesicles from the MSCs decrease proximal tubular epithelial injury. So the anti-apoptotic effects could be important. And then as I indicated, I think there's equipoise in the field between cell contact dependent and independent effects that explain the findings so far. So there's lots of unanswered questions. What are the contributions of the cell contact dependent and independent pathways to the beneficial effects of lung injury? And in line with that, what are the dynamics and the mechanisms by which the cells enter the lungs, stay in the interstitium, or transfer into the air spaces? How much of the beneficial effects of MSCs can be reproduced in vivo with paraffin products such as KGF and others? What is the role of microvesicles released by the MSCs? I didn't mention too much about this. Dr. Lee has an independent program at our group in which he's studying the microvesicles themselves for their repair properties. And then what mechanisms mediate the reprogramming of monocytes into anti-inflammatory phenotype. And of course, importantly, how are the MSCs themselves modulated in the presence of acute organ injury in vivo? And of course, will the MSCs be safe and effective in severe ARDS? And I just want to acknowledge that several different investigators who have contributed to this work. As I said, we lost Dr. Traber, who was really, without him, we would have never had the sheep studies. Gerard Curley has done a lot to help this program go forward, and a host of other individuals, including Dr. Lee, Dr. McKenna at the University of Minnesota. My colleagues at UCSF, Kathleen Liu and Carolyn Calfee, were terrific clinical investigators who worked with me on this, and a host of others. And now they're collaborators at Stanford, MGH, Pittsburgh, and Vermont for the clinical trial, the NHLBI, and then perhaps most important, the patients who consent to be part of our clinical trials and our observational studies, and the ICU nursing and respiratory care staff. Thank you very much.
Dr. Matthay, I'm sure would be pleased to take questions from anyone.
Yes, very much so, please. Yes.
Thank you very much. I'm a pediatrician.
Excellent.
So
is my wife.
I know her very well. But we deal a lot with viruses, and you skip the whole part of virus. There is some indication that MSCs depress the T cells and viruses. Have you done the ARDS study with the virus?
Yes, in fact, thank you for bringing this up. In fact, we just finished a whole program of using the PR8 influenza model, the PR8 model in mice, standard influenza model, and we find no effect whatsoever of MSCs on reducing lung injury and mortality in that influenza model in mice. No matter how we give them, we use both mouse and human. Humans seem a little unlikely to be beneficial with post-response over several days. And in fact, also Conrad Lyles and his group has just published a paper also showing that they don't appear to work in the mice in influenza pneumonia. We have some data that the viral load may go down a little bit with the MSCs, but you know, the viral load in influenza is only a small part of the story. So will they be beneficial in other viruses? I don't know, but I think we find no evidence of efficacy in the mouse influenza model.
What is the maximum period of time that the MSC can exert their beneficial effects, and do you think that a single treatment could reduce the risk of any subsequent cases of ARDS?
Yeah, the third question, what would be the duration of effect of MSCs, and would and would more than one treatment possibly be beneficial? So when we first talked to the FDA, we naively said, well, we'd like to give 3 doses of MSCs, day 1, day 2, day 3. They said, whoa, slow down. No. You can't do that in patients until you show us, Dr. Matthay, convincing preclinical data that more than 1 dose is safe. They said, we strongly recommend you begin your clinical trial with 1 dose. Study that. And if there were a beneficial effect in those safety issues, then you can consider advancing to a 2nd dose. I mean, I think I would imagine that a 2nd dose of 48 to 72 hours might be appropriate. We always worry when we're testing new therapeutics that we're not getting enough of the therapeutic agent to have a beneficial effect. But we have to go slowly here. I don't know how long the effect would be present. I know that Jared gave 2 doses in the rats, that you and John, you and Jared did that to get a sustained effect in their ventilator-associated lung injury model. It's hard to be sure. I don't think these cells last very long. I think that they are clear, that they're giving up their mitochondria, and I think they're not surviving very long. Most of the studies show their survival is limited. Armand, what would you say about their survival?
Yes, I would agree. We do have some data in the autologous setting suggesting that there is prolonged survival in the marrow. They do, in fact, lodge in the marrow and can survive for months. But most of the studies tend to use allogeneic cells. So there's this whole issue of immunoevasiveness that needs to be, I think, more rigorously addressed. But in the allogeneic setting, I would say that they do survive for a relatively short time.
Right, and this issue of immune tolerance, as Armand said, it's a big issue. There's no acute apparent reaction, but we need to learn quite a bit more about this. There is a trial, as you may know, that's in Canada with Duncan Stewart's group, in which they plan to get the cells to patients with sepsis, they're going to give the cells without cryopreservation. In our studies, as you could see, the cells are frozen, and then we thaw them. We give them without any DMSO, which is a separate story we found that DMSO seems to inhibit their beneficial effect in sheep. So all our studies were done without that. So we thaw them and give them just like we did in the human lung studies. But there is some reason to believe that the cryopreservation itself might impair some of the functions. So they're actually trying to do the studies at Duncan Stewart's place by thawing the cell, by growing the cells fresh in culture, and then harvesting them and giving them without cryopreservation, which I'm glad there's another approach. See what that shows. Yes.
Does priming or conditioning the MSCs enhance their efficacy as antimicrobial agents?
Several groups have started to look at that more. We looked, when I was on sabbatical in Paris 3 years ago, at the effect of hypoxia and also exposure to cytomix, TNF-alpha, IL-1 beta, and gamma interferon. And we actually didn't find that it changed the release of any of the candidate paracrine factors. But that is, I think, an open question that should be addressed. Tom?
I enjoyed that talk tremendously. I think it was a great talk that captures the spirit of translational research on the IMS. I mentioned before, I don't think there's a better lecturer that could come to give this kind of lecture. I went back and forth between basic science and impactful clinical trial design and some of the actual issues that an investigator has to deal with. I thought it was fantastic.
Thank you.
Now I want to ask you to do something different.
Good.
Which I don't think you did very much, which is to maybe speculate about the actual underlying biology here. What do you think the meaning, that's existentially almost, of taking bone marrow and putting it down the windpipe really is? Do you think that the normal hematopoietic system assists in the response to injuries in other organs? Is there any evidence for egress and migration? And if you think that, then do you think there's something wrong with that in patients who die in the ICU, or do you think there are other mechanisms or approaches that we should be exploring, trying to augment that underlying biology?
Well, I appreciate that very much. That is a deeper and more important biology question and translational issue. From a speculative standpoint, I think that these cells in the bone marrow have probably evolved over eons as guardians of the hematopoietic elements. And when we discovered these antimicrobial properties, I thought, okay, that actually makes sense. What was the greatest threat, is the greatest threat in evolution to the host? It's, other than trauma, it's probably infection. So it would make sense that you might have a cell in the bone marrow that would have antimicrobial properties. Since there were no antibiotics available to the dinosaurs, it makes sense that these cells might have evolved to have antimicrobial properties to protect against, protect the hematopoietic elements. Similarly, you could imagine that the anti-inflammatory properties, the immunomodulating properties, could be protective for the bone marrow. So I thought that might, in fact, be why the MSCs in the bone marrow have those properties. Now, it's a terrific question, of course. Are they mobilized from the bone marrow? And to the extent that they can be mobilized to an injured organ, the kidney or the lung or elsewhere, couldn't that be a major determinant of who does well with acute organ injuries such as ARDS or a kidney injury? And we just don't have yet the markers to quantify their ability to be mobilized, I think, yet in a experimental model, or yet in the patient. But if we could track these cells and their accumulation in another organ, and what influences that, we know that a lot of bone marrow cells are mobilized under different conditions, of course. So it only makes sense that maybe some of these MSCs, to a variable degree, are mobilized. So that could be a big determinant of the progress of organ injury. So I don't know. Does anyone else have ideas on that point? It's a terrific fundamental idea to to be able to measure their egress and movement to the injured organ.
Peter over here.
Yeah, we just wanted to comment on the concept of molecular imaging of radiopharmaceuticals or so on. I mean, you call it a therapeutic, but it's very different from the traditional therapeutic drug, where it usually exists in one or a couple of metabolic states, and we have a good idea of how it's distributed, and we can track where it goes. Whereas as soon as the cell is injected, it's transformed. And then probably each cell is in a different state. Each mitochondria in each cell is a different state. And it's very hard to really treat that as a simple, instrumental molecular drug receptor interaction type.
Right.
We think of when we talk about therapeutics.
Right.
So, and it's more existential than it.
Yeah, it's so much more we don't know than we do know. And yes, we've done GFP positive tracking of the cells, over a few hours and over time. We can find them in the spleen, lymph nodes, like others, but it's not terribly quantitative. It doesn't tell us much about cell function. It tells us nothing, really. And so this is where, in fact, although we're all for translating work, you can certainly say, well, wait, we don't understand this well enough to actually test it in patients. And so this is where, to be honest, I have my own uncertainties whether we're proceeding too quickly or not. It's unsure whether we know enough to proceed. Now, there's another angle to this, of course, that is obvious, I think, to everyone in the field, which is that could you use as a therapeutic the conditioned media as opposed to just the cells? Let's give up on the mitochondrial transfer as a critical mechanism. Why not use the conditioned media itself? Might be safer. Maybe you could quantify most of the key products. We are very interested in lipoxygenate 4, which appears to be a major product of these cells and has anti-inflammatory properties. But maybe you could actually quantify this, but then you'd have to really do it. And the FDA won't let you go forward. Ironically, they'll let you go forward with the cell-based therapy, where there's all this unknowns. But if you come in with the conditioned media, they're going to say, oh, wait, now hold on. You've got to tell us and show us that the major products are stable under all the conditions you said, and but still, nevertheless, I feel like there's a potential future there. There's several commercial groups now in the market. They're working on producing MSCs in efficient ways that will be less labor intensive. And it could be that as the future unfolds, that conditioned media might be better, including conditioned media that's been in which the cell's been treated X or Y way to upregulate certain products. And then, obviously KGF has been a major player here in a lot of what we found. So we are in the process of doing a phase 2 trial for ARDS with KGF being run by Danny McCauley in Belfast, because I think, it's good to have a dual strategy. So sometimes work like this will give you an idea about a therapeutic, even if it's a single.
So we'll need to stay on time. Dr. Matthay will be available during the break and at the panel discussion as well for other questions if you wish to ask him. So I wanted to thank you on behalf of IMS. Echo Tom's comment. What a spectacular example of translational research. This is my model of what translational research should be. So I commend you on your science, but I also commend you on your person. I met Michael last night when I picked him up at the hotel. Within 10 minutes, we were showing each other pictures of our grandchildren. And I was really struck by your humility and your personableness. And you tied it all up together and made me realize we really are soulmates, not only growing up in the 60s, but being members of Red Sox Nation. I mean, what a better... Couldn't have had a better choice. So on behalf of IMS, I want to give you a token of appreciation from all of us, and let's thank Professor Matthay. Thank
going to move to two presentations, but let's